going to just start with the, thank you, rather noisy. I'm going to start with the antibiotics. They put in a new computer. I am beyond happy. I mean, they actually have a high def cord that I can plug my laptop in when we use it. They have the new thing here rather than that cumbersome idiocy. And there's like a new, just, I'm beyond happy. Beyond excited. So this is I'll show you where, scroll down. When you get just above where it says week 4, I started to pop these things in, I think on both of these. So I have an outline for antibiotics, which I did years ago, and we will, this is big. Boy, I told you the first exam that they had, the nursing students who were a year ahead of you had, was all about antibiotics when they were doing their pharmacology course. That whiteboard was covered. So I thought, give you guys a head start on this, and then we'll talk about, but I really think it's a very important therapeutic chapter. And it's very, to me, it's very, very interesting to teach, because I know a great deal about that. I mean, when you're someone who's operated on people, you learn a lot about infections. When you're someone who's worked with diabetics extensively, you know a lot about infections. Infections are part of the problem, a large part of the problem. But nevertheless, that's kind of where we are. And I'm going to start with this one. I loaded the one that's in your textbook, which is, I believe, chapter 10. There's a lot of useful information. I'll flip over to that. I'm going to start the introductory nugget here with this chapter 20 from the Nestor text. For whatever reason, I have an older edition. It doesn't matter. But I'll find a newer edition. That's sort of incidental, but I will get that looked up immediately. Nevertheless, this guy, Ehrlich, just a little, and I like because they give you a little, historical things. There was a movie about it years ago called Ehrlich's Magic Bullet. Okay, many years, like 1940 or so, and look what you're doing in the lab. Okay, so he looked at bacterial. Some dyes stain bacterial, but not animal cells. So he knew they weren't the same cell, and he wanted to find something, quote unquote, the magic bullet that would kill the bad guy and not hurt you. And he worked with, of all things, arsenicals. Arsenic is a famous poison. It's really the original rat poison. It's been around forever, a naturally occurring product, and it was used to treat neurological disorders. People had tics and were having, like, the nerves were unstable. Okay, back in the day before they knew anything about modern medicine. So he began to tweak it little by little, and the 606 test. And what he was trying to do was treat syphilis. Syphilis is still around today, legendary, been around forever, literally, since modern man started chronicling this stuff. What happened was, in late stage syphilis, people began to go crazy. Sanitariums. We'll talk about syphilis again when we do genitourinary disease. We hear about someone going into an insane asylum. It was for syphilis for the most part. Early on, because it destroyed their brains and was passed on to their children, and there's all sorts of stuff that goes on with it. So he tested all of these compounds, and at least he saw one that was at least effective. Its fancy name is arsphenamine. The fancy title, I have this just historical contact, salvarsan, which meant salvation through arsenic. Okay, it was real, but what it did, okay, prove some chemicals could selectively kill. And that's really the basis of the start of this. It's a notion that's really important called selective toxicity. These are definitions that are important. It means more harmful to the microbe, the bad guy, than the host, us. There's several definitions early on. Optimally, the perfect agent, and there's none, would be absolutely able to destroy the invading whatever it is, microbe for a generic term, and leave us completely untouched. That would be it. And there's no perfect agent, because you're going to see either have trigger your immune system to make allergies, or do something bad to your some other insides in some way, shape, or form. We call them side effects. They are not interchangeable terms. And we'll get to all the terminology, but this is a big player. Okay. So we'll look at the definitions that are associated with it. So I mean, these are colorful. You're doing this. You're coming back. That's why I wanted to do it, because we're going to talk about it, right? You're going to see these tests. They're doing the same tests forever. Those Kirby Bauer plates. You're looking at them on whatever, tomorrow, Wednesday, correct? That's a way we can identify. It's not a guarantee. You're going to see, you're going to class. They have a little teach on that, how you classify them and measure them. SI and R, sensitive, resistant, intermediate, not yet. It's a what we call, just to give you an idea of why it's important. It's what's called a qualitative test. The fact that something has a bigger zone, look over here, than a littler zone doesn't necessarily mean it's better. Okay, it's really it's qualitative means yes or no. Do I feel better or not? Yes or no. I'm not sure. Those are really the three answers. Quantitative means you have a number. It's different. So those are important. Those are definitional things as you get going with your careers. So it's that stupid thing they ask, on a scale of 1 to 10, how much pain do you have? I'm good with mild, moderate, and severe. Always have been. Okay, it's like, oh my God. If you grab it, it's a 10. If you leave it alone, it doesn't hurt. I'm sorry. Pet peeve. So moving on. We had really very little of these till almost 1940, but I'll give you the history lesson. So common diseases, pneumonia, staph infections, before penicillin. Penicillin was actually accidentally discovered, as you will see. At least it was written about, I suspect it's a little before this, in 1931. Understand, penicillin comes from moldy bread. Penicillin is the most common mold in our environment, and it loves white bread. The green stuff, blue-green stuff you get on the white bread sometimes, starts white. So people were using it. I mean, inadvertently, we used this. Aspirin came from willow tree bark. Okay, people made infusions and poultices and a variety of things that were using things that we use today, except we have refined them. And we continue to refine them. That's the interesting part about this. Okay, but they didn't have a whole lot to do. You could bleed people, you could open it up, you could drain it, and when it didn't work, you had to cut off the part. So, and this is the problem, why it's such a darn important unit, is we have misused them. We have forgotten, and I'll say this again and again, the basic tenet of, oh God, I forgot. Tell me I saw a ding, so we did start. I'm sorry. I'm going nuts. First day, there it is, first day back. The basic tenet of really what the idea, they thought, oh, these things, if we kill them, we're done. But what did they use? They hammered the heck out of them. In the PowerPoint that accompanies your textbook, the Cowan textbook, they identify it early on. It's called shotgun therapy. We heard about that. We were just learning. Shotgun means I'll use it. Somebody get an infection, I'll take the strongest agent I can because I want my base to get better soon. And we clobbered organisms. But the trouble was they got used to being clobbered. And it's a living thing. What do? It's chemical warfare. Things that aren't killed adapt, overcome. That's what evolution is. They develop weather-induced mutations. That's why you do the genetic unit first, or acquired mutations, or transfer things that bacteria can do and other organisms can do to a certain extent. And what happens, the ones that survive effectively become resistant the more you clobber them. And in the meantime, you're killing all the good bacteria that's so useful to you. And the major problem when we use antibiotic therapy, okay, gastrointestinal problems, the major side effect is GI distress. Nothing is even a close second. That's why people, it gives me diarrhea, I vomit, I feel lousy, I feel like I'm going to throw up, my head is killing me. All of those things are more or less gastrointestinal. And yet we continue to do this to the point where we have evolved, and I've said this before, we have evolved Staphylococcus twice. But when 90% of it was sensitive to penicillin, back when we started using it in 1940, and in about 20 years, we found probably 70 to 90% were now resistant to penicillin. They were producing an enzyme to break it apart. We changed it again, didn't change our tactics, still hammered it, and all of a sudden we got MRSA. And MRSA was showing its ugly head when I was a student. That's almost 50 years ago. Now it's everywhere. We don't, whatever that old saying is about, if you got to learn from history. If you don't change what you're doing, you're doomed to repeat it. We're finally figuring it out. Unfortunately, we're running out of antibiotics. And that's just so inducing mutations. So ends the sermon. Ehrlich, Salverson. Here's another one. Great agent you're going to find. You're going to use a variation of this agent all the time. Believe it or not, for what we call community-acquired MRSA. Something you're all going to see, whether you're doing it in healthcare or somebody in your orbit has it. Okay? There was a red dye that they were treating strep infections in animal called Prontosil. They usually get dyes from berries. So in test tubes, it didn't work on bacterial specimens, but enzymes in the blood split it apart and created a drug they named sulfanilamide. This was the start of sulfa drugs that we use today. So now, and we'll learn about it when we go into the science behind it. Sulfanilamide came out was extraordinarily because we had it. Bad guys in World War II did not know it. Soldiers carry around big packs. Somebody goes, you're in the field, no choppers, nothing like that. Or those field hospitals, you got to carry people with litters. And this, I mentioned this before, a couple of rifles, a couple of ponchos, hope they get the job. We saved a lot of people because in that pack they had green powder called sulfanilamide. We sprinkled it on the wounds, really stopped tremendous amounts of soldiers losing limbs, parts of limbs to it. It was a big, one of the many things was penicillin that allowed the Allied forces to win World War II. They said, wars are wars of attrition. We kill more of them than they kill of us. We have more people to show. I hate to say it like that. That's pretty bad. Moving on. So what we use today, and many of you will know people, and you yourself have used it, use drugs that are a combination of sulfamethoxazole, with another one that's kind of in the family. You're not to write these down because you can see it. Called trimethoprim. It's marketed under the trade name Bactrim receptor. They're the drug of choice for relatively common bacterial bladder infections, mostly in males. Very common. Because of problems that sulfa drugs had, a common complication of them. Okay. We stopped using them. They were great because they worked. We didn't have to give big doses. And nowadays it's cheap as chips. It's very inexpensive. Magic. We found that for what we call CA, community acquired vaccines, MRSA, the first choice is typically one of these. It's some big powerhouse. They're charging $1,000 a dose. Something that your copay would probably be $1. Giant used to give them out for free. And you've become very familiar with that. Kind of gives you. We went back to it because of the complication. People started used. We got to use more modern antibiotics, and they said we're running out of stuff to treat MRSA. And magic, look at this, it worked. Did anybody use something called nalidixic acid or triple sulfa as one of your those little circular papers? Is that ringing a bell with anybody? You probably used ampicillin, penicillin, tetracycline, the usual culprits. Nalidixic acid and triple sulfa are one of those. So I taught that last while. So chemicals are used to treat disease, antimicrobial drugs or antimicrobials. So the parent name is, and notice the term chemotherapy. It doesn't have anything to do with cancer. It means using chemicals to make people better. Antimicrobials, that's the global term. Antibiotics are different. You'll see the definition shortly, but they come from microorganisms, and they're produced, as you might remember, in that stationary phase of the growth curve to either kill or basically stop or inhibit the life cycle of other microorganisms it's competing with. That survival of the fittest rears its ugly head again. So Fleming, I hate to say it, if you had done this in lab, you would have gotten F. He contaminated a culture of staph. Remember they say no food in the lab, that thing? He probably had some white bread and crumbs of it on something. It got on his lab plate of staph. When he came back after the weekend, he sees a big blob of mold growing on it and no staph growing around it. And he's like, that was good. Honestly, this is exactly, you'll see that you can find the pictures anywhere of this boo-boo. Okay. He named it penicillin because penicillium was the name of the mold. Okay. Killing a lot of things, almost all gram positives, by the way, a few gram negatives. They asked him to purify it, couldn't do it. Like I'm telling you, you have to understand, when it comes to doctors and scientists, there are those at the top of the class and those at the bottom of the class. As long as they help you, they're good. It doesn't even mean they're all brilliant. So notice these two gentlemen purified it in 1941 to treat a police officer. He improved, but they didn't have enough. So in World War II, Brooklyn, New York, Pfizer, the largest pharmaceutical company in the world, who brought you the Z-Pack, who brought you Viagra, the largest pharmaceutical company in the world, dedicated all its efforts at its main facility, I think its only facility in Brooklyn, in New York City, to making one agent to support the war effort. And so we had it. The bad guys did not. Just to give you an idea. But this is the problem. This is like its family tree. You see, we didn't even know we're doing this is like genetics. We're doing these are like pedigrees. Must have been crazy in the family. They all started here. And because it was so effective, they began to tinker with it. And over the years, they tinkered with it because it worked well. Why did it work well? And it didn't have a lot of side effects. Because how does it work? We already went over that. Very first week, it attacks the one thing bacteria has that we don't. Peptidoglycan. It doesn't let it assemble. If you can't assemble it, bacteria can't replicate. They all die. There's a great term associated with that. The most important term in deciding what antibiotics to use called bactericidal. You're doing those tests, okay? And your Kirby Bauer plates. The ones you're going to list them. The ones that are what we call sensitive, which means they work. Which one do you choose? The first thing you look for is bactericidal or bacteriostatic. I want something to kill the infection. First thing we look for. We learned about antibiotics and prescribing and I want one that's going to kill it. I do, because what happens is, it's a very important word. When you use something called bacteriostatic, it slows. Remember this for antibiotics too, you can kill it or slow or inhibit the growth zone. Okay, at that point, that's making the assumption. You know what happens when you assume nothing good, that your immune system will take over. I want it dead. More. Sleep with a fish 6 feet under. Dead. Deceased. Taking the dirt now. Okay? So, all of these things are there. And what they did is they expanded it. They saw it didn't work well on gram negatives. So they started making things that we call ampicillin and amoxicillin. These were penicillins where they broadened the spectrum. to make sure it could attack gram negatives. Okay, they found they needed things to combat a lot of these enzymes of Staphylococcus. That's where methicillin and oxacillin, etc., came about. They even had another tree over here, and that tree, and you're not going to have to know all these names. Ones you'll know, I will tell you about. On that side of the tree, what do they have to know? What do they have to do? This is called extended spectrum. That's because we were trying to address particularly resistant forms of gram negatives that we have very little answers to treat. They are a big problem. We will get to them. They have a fancy name that you'll have to learn about called ESBL. It stands for extended spectrum beta lactamase producers, and we'll get there. Don't let it. There's a lot here. This is a big chapter. We might as well get to it now. Okay, so interestingly, they began to. This is why we constantly sample the soil. They were fortunate, bless you, that penicillin was basically in the air all over the place. But then they said, it was only working for gram positives and limited gram negatives that were easy to kill. Remember the term fastidious, syphilis, gonorrhea, very easy to kill. If you can't grow them very easily, they're very easy to kill. If they're like E. coli and grow easily, they're very hard to kill. And so Waxman, and this was the name of this very important species called Streptomyces. This is a soil bacteria. Okay? And that's where the name streptomycin comes from. And not only from members of this particular group, the Streptomyces group or genus, came a ton of antibiotics. Erythromycins, which is really the Z-Pak that you've heard a lot about. Tetracyclines are coming from a similar area. So that's why we're constantly sampling areas where we haven't been in the soil to try to identify antibiotics. Well, Selman Waxman in the early 50s was able to identify this, okay, and they're constantly doing the soils, and he got a whole new category he named for it called streptomycin. We typically use the suffix mycins for indicating some kind of antimicrobial. Not all. So in the old days, tetracyclines were called terramycin. Okay, you're going to hear more about all of those things as time goes on. The streptomycin. So all of this, and interestingly, its target was basically gram negatives. It's got a fancy name, and we study them by categories. By this time, you should be annoyed sufficiently with me about knowing things not by name, but by categories. Categories are important. It's like peroxygens and that stuff. Don't yell at me. Be happy I gave it you. It's okay. I'm having a day. And it's early. So we're going to look at the categories. Here's selective toxicity. And here's a term. The terms are identical. They're just different. So selective toxicity, easy, more harmful to the microbe than the host. And we're going to look at bactericidal B or versus bacteriostatic, doing the antibiotic, antibacterial agents first. So, for instance, human reason. So they talk about something called the therapeutic index. How this is calculated, we call it TI. And if something is therapeutic, it has to be greater than 1, preferably substantial. It is a ratio. So we look at it, we consider the average lethal dose of an agent. This applies to any medicine. Well, 50 at the 50th percentile, or LD 50. And we have another measure called ED, or effective dose. The same percentile. Because half the people die and half the people find it effective. So always put the lethal dose up top. So for instance, if the lethal dose is 90 milligrams, and the effective dose is 10 milligrams. That's a safety. You pull that therapeutic index of 9 to 1. The higher the index, the safer it is. Why don't we use alcohol for an anesthetic? Alcohol, the lethal dose, and the effective dose would be a general anesthetic are very close. So you're almost as likely to kill somebody by, and that's what they did in the old days. They had to drink a bunch of alcohol to operate. That was just part of the problem with alcohol. So other influences. But nevertheless, this is an important thing. The therapeutic index of any agent is considered therapeutic has to be greater than 1. And the larger it is, the better. Another term that's synonymous with it is safety margin. The margin of safety, because you never know, well, the doctor gives me these antibiotics, I'm feeling a little better with 1, I'll eat 8. You got a whole bottle of 40, well, let's take them now, get better. I want to get better next one tomorrow. I don't like that. You know, teaching. Safety margin is the difference between the 90 grams, just subtract it. So it's 80 milligrams. It's just another measure, sort of like the absolute value, how far apart they are. Good. So that's what those are terms you're going to see, and I'll ask them, but believe me, in nursing they're going to say, "Oh, which one has a higher therapeutic index?" Maybe they'll want you to do the calculations. I try to stay away from the calculations. You guys don't like me as it is. This will only make life worse. I have missed doing this. I'm energetic. I only have another 7 weeks to go. Ooh. I'm going to have, because of something, there's a potential I'm going to have one of my colleagues in to watch me teach. We have to do these like evaluations periodically. You need to be on your best behavior. Do not, and I'm speaking to you two over there and your erstwhile buddy who I know is not feeling great this morning. Don't mess around.
Okay, fine. Listen, don't mess around. I'm trying to show him how my computer's giving right now. I don't know what's going on.
It's okay. It's okay. You're not out of it yet either. Nor that well. Not even talking over here.
Tell Yoni not to come later.
What?
I can't talk till Yoni not to.
No, you didn't do anything yet. But we're waiting. Again, so penicillin G, high index, interferes with cell walls. We don't have them. Okay. And a lot of times, why do we use things? Why do we put certain things in topical antibiotics? Because your body would be destroyed by them internally. Doesn't mean they're not good. Again, that's what they did. They did that here in Pittsburgh. They took 3 agents that almost were impossible to tolerate internally. One was a cousin of that streptomycin called neomycin. Another one is bacitracin, which comes from that B sub that you work with in the lab, Bacillus subtilis. And another one is called polymyxin B, which I'm not sure where it comes from. And those were so harsh internally, they put them together and called a triple antibiotic or Neosporin. And that was developed here in Pittsburgh. Just a little historical Pittsburghian stuff. I don't know why I'm so enthusiastic. I'm transplanting for the last 50 years. Basically a New York metropolitan kid. Bacteriostatic inhibits, but your defenses have to go to work. Okay, bactericidal. Okay, sometimes they're only, but I'd rather go in that direction. The deciding factor. Spectrum of activity. So we're doing the definitions, as you might have guessed. Why do I have another prescription of it? Giant Eagle, very annoying. Broad spectrum antimicrobials. The whole, and this is what I talked about, shotgun therapy. They took antibiotics that were massive and used them for everything. Okay, if it's life-threatening, sometimes you don't have a choice. A lot of times we still learn when I tell you about it. We start broad and narrow right away. I would tell patients I would take a spear or a specimen. I'd either look at it in the office, fortunately the hospital lab was right there, and I'd say, based on where it was and aspects of the infection, I would say, okay, I'm giving you a prescription for this. This is a 5-day supply with a refill. There's a chance I'm going to know in 24 to 48 hours this isn't the right prescription. I said, but I do need to give you something right now. We call it empiric, EMPIRIC. So I made them aware that this is my best guess now. I hate to use the term guess, but I don't have any tangible, I don't have a whole lot of empiric evidence. things I can observe. No, it's a diabetic. No, it's not, etc. History. And I'd say, okay, if you don't hear from us, get it, call if there's any problems and blah, 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 blah. But if not, get it refilled and we'll go from there. If we call, you may have to get another prescription. Nobody likes surprises. Not when you're treating people. You may have to have the same spiel depending on what you're doing. Okay, so broad spectrum when you don't have time, and then you narrow the spectrum. Okay, I know what this is. I know what strain of Staphylococcus this is. I know it's Streptococcus. I know it's a strep throat. Now I can use the right agent and avoid disrupting the normal microbes that are your buddies. All those wonderful gut bacteria that we're so desperate to have. That's why we have prebiotics and probiotics. We're aware of this now. Narrow spectrums, very limited, but you must know exactly that's the test you're doing. Kirby Bauer is all about finding the best agent for that particular one with the least problems, preferably bactericidal than bacteriostatic, and less likely to do damage internally that's unanticipated, cause side effects. Sometimes we have to use combinations. I don't know why. And when a person is taking multiple medications, and sick people do, you have to make sure they don't combat each other. What I showed you there on the board, this trimethoprim and the sulfamethoxazole, okay, those are synergistic. Together, they work better. And I'll show you the pathway. It's a great pathway because, like penicillin targeting peptidoglycan, it targets a pathway that bacteria have that we don't. That's a win. It has to do with something called folic acid, which we have, which is kind of a B vitamin that we have to acquire through our diet. We can't make it. We stop bacteria from making it. It helps us assemble DNA, RNA, and stuff like that. Those A's, T's, G's, C's, and U's. You've heard so much about it and are so excited about it. I'm not seeing waves of enthusiasm here. I'm seeing tired. You spring breakers. Hope you were not down there in all those places doing all that stuff. I'm not asking. You know what they say about don't ask, don't tell? I'm there.
I'm there too. Great.
I'm there too. You've seen my daughter. Don't ask. She sent a video. I don't even know what I did with it. They were doing a party at this club they go to on the south side of Cabo that they almost bought. And she's like, they're doing some mega party for like an anniversary, and she's jumping off the thing. And she's seen this woman who's now 40 years old still. I have been what I've been through. Moms and dads, give me a call. I survived. They will too. Nevertheless, that's synergistic. There are ones that are additive. I mean, I don't want to go crazy with this, but I'm just trying to give you an idea. That's not something I typically ask. Other things, tissue distribution, metabolism, and excretion. It depends. Not everything crosses the blood-brain barrier. We can't use everything to treat bacterial meningitis. It's very, very specific and extraordinarily important to treat it immediately because you don't have any wiggle room with bacterial meningitis. None at all. And so, but you have to use an agent that will cross the blood-brain barrier. That means it can get through the membrane because it doesn't get through those neat little openings. We talked about that. Some unstable at low pH read that as stomach acid. And so typically the difference between ampicillin and amoxicillin, ampicillin was effective in pill form, but not great. That's what I took as a kid. A few years later, they came out with amoxicillin. That's what my kids took 15 years later because it's more active orally. We'll go over a chart that talks about that. They tinker with it. That's the difference. They work, they're basically the same, a subtle tinkering with the chemistry in the lab, and we call those semi-synthetic. Semi-synthetic means it's naturally occurring, but we play with it in the lab. Penicillin G would not be tinkered with. It kind of came directly from it, as opposed to adding things and tweaking it. And you'll see how we do this. Okay, so they have to be injected, and it's much easier to get people to comply and take it orally. Half-life. People don't understand. Half-life is how long is not a time it takes half the dose to be extreme. Okay, rather to be broken down. So half-life kind of means how long it stays in your system. That's all it really means. So people, when they had the Z-Pack came out, oh, I only take this for 5 days, it's stronger. It's geared to be released slowly over time. So effectively, they've tinkered with it in the lab and made it more, have extended its half-life. So those 5 days of doses will actually last for 10 days. Okay? We look for things like that. Things that have a short half-life, you have to give them to them 4 times a day. Try to get somebody to take an elderly patient, you have to take 2 of these tablets for 10 days, 4 times a day. That's why we like to have medicines you take once a day or twice a day, because they're less likely to forget. And even if they have them and you go and you arrange those neat little, you know what I'm talking about? Those plastic containers for grandmas and grandpas. You're a popular great grandpa who still remembers everything. Be able to do that. So that's important. And look at that, kidneys and livers. You've got to adjust the dose for them because that's how we detoxify. So when we talk about an adverse effect, okay, they are different than side effects. Allergic reactions and toxic or side effects. Toxic because they can kill you. Both of these. Allergies are the direct result of triggering an overly exuberant response of your immune system. Are you guys doing immune and lecture finally? Do you know your T cell from a B cell? Oh, you're having fun with that. Nevertheless, that's what an allergic reaction is. You take the dose one time, and any time after that, after the initial dose, your immune system can go. That's what we call it. There's the one that can kill you is called anaphylactic shock. Nowadays, we also use the term anaphylaxis. Terminology was updated to be a little less confusing. And anything that you take that gives you hives, a rash, difficulties breathing, swallowing, swelling, that kind of stuff, that's an allergic reaction. That is not a side effect. And you're going to run into this. Many of you with patients are going to come, oh, I can't take that. What medicines are you allergic to? Oh, I can't take that. Just tell them you're allergic to this and allergic to that. They won't give it to you because it upset your stomach. That is not an allergic reaction. It's a side effect. You can deal with side effects, and so they won't kill you. But allergies are a whole lot trickier to deal with. Sometimes it's a must to use potential allergy things in a therapeutic sense. When my dad died, he had in old days, we injected iodine dyes and did x-rays, basically to see. We have much better technology today. He was allergic to the iodine, but they had to they were going to do heart surgery, so they had to do that for his heart, called an angiogram. And he told them they were allergic. He said, look, this is more important. We'll deal with the allergy. Gave him Benadryl and stuff like that to sort of pre-medicated him with that. Fortunately didn't have a response. It wasn't a successful surgery, unfortunately, but that's a sad story. But you get the idea. So you can deal, in certain situations, you have to understand this. I'll tell you some anecdotes about that. Like I said, I've been doing this. I did this for a quarter of a century, trying to find the right. I know it seems like a long time for you guys who basically aren't a quarter of a century old yet. But understand that's why I hope to be of benefit to you. So adverse reactions. So this is the big one. And there's a classic reason why C. diff is such a pain in the neck. Broad spectrum antibiotics. They kill the ones that are easy to kill in your gut. And then all of a sudden this endospore former that's there, and a fair amount of us goes, oh, okay, my turn. If you never had C. diff, you don't want it. So we'll talk about that when we get down to what's with C. diff and the metronidazole and things that use that. Properly used and prescribed. Resistance. This is where the genetics comes in. Some things are innately resistant. Mycoplasma, walking pneumonia, doesn't have a cell wall. Cell wall agents like penicillin don't work. Why bother? Okay, innate or intrinsic. Gram negatives. Basically, most penicillins, unless they're very easy to kill, don't work. Okay, that outer membrane. Again, there's a drug you're going to learn about that's one of the special drugs that we use for MRSA called vancomycin. It doesn't work on gram negatives. Period. End of sentence. It can't. Nothing that's ever been done to it will get it through that membrane. But we still have to use it. At one time, and even now, it was considered the drug of last resort. I'll tell you about that. Acquired mutations. Acquired all those transformation, transduction, conjugation, transposition stuff I talked about. That's why I had to give you that context. You saw that take-home test. Can you imagine having to do that in class? The time allotted? It's pretty hard, I think. Most of the students are. So if I had combined that with some other stuff, normally I add those first 15 questions to the exam you took before you left. None of you would want to even speak to me again. Most of you don't. By the way, when we have a visitor in the class, you need to be interactive. Ask questions. I know this is a nuance. You would not. Yeah, you got to spread it around. I have to actively engage all of you guys.
We'll go from this side of the classroom to the other side and then jump back.
They'll know. They're my friends. This is an array, as we conclude this introductory material. This is where they are all working. We have targets in the cell wall itself. The vast majority, thankfully, of the ones you're going to have to know either attack the cell wall, no cell wall, it's unstable, it bursts, or they attack protein synthesis somewhere along ribosomes and how they work. Those are the giant categories. Okay. The trouble with cell membranes, the cell membrane in bacteria, remember I mentioned in passing polymyxin B, the trouble is it attacks our cell membranes. That's why it's very, very rarely used. We'll talk about daptomycin. Okay. Here's that one, sulfonamides and trimethoprim, the one I put up there, the folic acid pathway, or folate biosynthesis. Only bacteria, they're good. That's why these guys are very well favored. We all do protein synthesis. That's where the problem is. Remember that endosymbiotic theory? Our ribosomes have 70S. We have 70S ribosomes rather in our mitochondria. Unfortunately, when we attack the 70S mechanism here, it also affects our cells. That's why there's more side effects. There's nothing, again, is what we call a panacea. Ain't nothing perfect. This is a very interesting category for fluoroquinolone. It's got a fancy name. It's got really a couple of wonderful agents in there. Did you put any tabs on there with something called ciprofloxacin? Cipro? So, I would have forced you. Here's it. We're almost done. Summary table to look at, what we have to go over. There's what to talk about. We'll start here when I see you guys on Wednesday. Thank you. A lot to digest, as it were.